on this topic, digital microfluidic biology. It's design automation and fault management. This particular topic in this lecture will be discussing about basics of this digital microfluidic biology, some of its introductory part, its evolution and design automation challenges. Design automation challenges, fault detection issues, and design goals. So digital microfluidic biology is a recent research trend and it has evolved around 2000. It has evolved around 2010 mostly, and prior to that the research began in the period around 2000 and all. So it is digital microfluidic biology concept is evolved over first generation of biology. Mostly it is familiar as a lab on chip devices because most of the pathological and biochemical experiments with minimum requirement of large samples and different samples. Capable of performing different clinical biochemical experiments, pathological experiments mostly which we say in the lab, pathological laboratories. Other application, it includes DNA sequencing, air contamination detection, different protein analysis, drug discovery, so many applications it has. So, in today's lecture, we will see about its basic different complexities that we need to take care of during operating with the research of digital microfluidic biology and some of its research scope that we will introduce later on. So if you look into the view, since this is a very new topic, normally the first one in the top left corner, it is the continuous flow biology, the first generation biology, and which probably could not manage many operations. The later version, that is the digital microfluidic biology, the real view of the digital microfluidic biology is given in the bottom right corner. So if we look into the differences in between these, so the first generation it was continuous. So the first generation it was continuous fluid flow mechanism based. The next one is based on the digital microfluidic biology. It is based on the operations electroweighting of dielectrics. We will see how the electroweighting of dielectrics occurs in the next slide. In the first generation biology, the limitation was it is capable of only performing one particular experiment. So there, one single experiment, single biochemical experiments can be done. Whereas in digital microfluidic biology, we can perform the parallel bioassay operations within the chip. In case of the first generation biology, if we look into the components of the first generation biology, it was a little bigger in size. So it took probably the larger space for the assimilation, micro level components, and only we can attach single level of preservers in the first generation biology. Whereas if we look into the second generation biology, which is the digital microfluidic one, DMFT, over there we can attach multiple preservers, multiple source components, and it helps in performing multiple parallel operations together. Obviously, the first generation biology was slow on the operation. The second one, the recent version, is the first enough. First one was fluid based, and the second one is the droplet based. We will look into the details what we mean to say by the droplet based mechanism. And already, as I said, that parallel operation is not possible in the first generation, whereas in the second generation, passively parallel operation we can perform. Now, if we look into the different steps related to the simulating different operations in the digital microfluidic biology, starting from the concept of different bioassays. So we know the biochemists were working in the different pharmaceutical companies and different laboratories. So these biochemists probably they provide the concept of different bioassays. What are the different reagents required? How we can perform different reactions within that, and what order, for the what time, what volume of requirement, in what ratio they need to be combined together. So all these biochemical reaction details we gather from the biochemist over there. Now according to all these details, next the task of the computer scientist in combination with these biochemists is to decide the bioassay protocols. And these bioassay protocols, in our terms, we say this is the behavioral simulation. In what sequence this protocol will be mixed, in what distance and what time we need to maintain. So all these details is designed in a behavioral model, and probably one of the VHDL tools are mostly applied over here for designing this bioassay detail. Next step, we move on to the architectural level synthesis. In the architectural level synthesis, mostly we try to attach different dispensing units with the chip, and these different dispensing units, when they are attached with the chip, and this probable attachment is done along with the module placement and keeping in mind about those different bioassay protocols. So this is a dynamic operation, and with respect to different bioassay protocols, it is decided that where to attach this different dispensing unit with the chip. Then comes the task of design automation. So in detail we will be discussing about the design automation challenges, mostly the droplet routing protocols and different things associated with the droplet routing protocols. And then the final stage, definitely there is a fault management, because extreme level of precision and correctness is required for these sort of operations, which associated with different pathological operations, biochemical reactions, and also we need to be very precise enough. So fault management is an inevitable part of this whole process. So now, if we look into, earlier one was a real picture, now if we look into the schematic diagram, so this is the left diagram, it represents the schematic layout of the digital microfluidic biochip. Specifically, it is fabricated on two parallel glass plates. The top one is mostly the carries over there the ground level of voltage, and over the bottom one we try to fabricate micro array of electrodes. You can see in the left diagram, it's a two-dimensional micro array of electrodes. Over there you can see it's a 6x6 structure. Some of the dispensing ports, as per the optimization process, it is attached with this micro array structure. We have optical detectors. Through these optical detectors, we try to detect the results of different biochemical operations. So mostly these are the light sensors, and the intensity of lights and different types of lights are used over here to find out the results of these biochemical reactions. And the photodiode, which is attached in the another end, mostly at some point of time, it checks the operability of the particular biochip electrodes, how they are performing, whether they have met with any particular fault or not. So for that particular purpose, the photodiode is attached. Now we were talking about, like, the first generation was continuous flow based, and the second generation is the microfluidic operational based. So in the microfluidic operation, in the second generation of biochip, mostly we use electroweighting of diodes, dielectrics, and apart from electroweighting of dielectrics, there is another phenomenon on these droplets that is called dielectrophoresis. Now dielectrophoresis is not mostly used because of its operational complexity. So majorly we use electroweighting of dielectric. So over there you can see in the B diagram, the micro array of electrodes, each of the cells, they are separated with a little bit of spacing. These spacings, it tends to 0.4 micrometer or to 0.2 micrometer, these spacing. So little bit of spacing. So from one particular electro array cell, we need to move droplet from one location to the next location. And in this particular process, we need to keep one particular cell where the droplet is residing with the lower charge, and into the adjacent cell where we want to move the droplet, that particular cell will be supplied with a higher potential. Then the droplet, due to its electroweighting tendency, it will shift from the lower charge region to higher charge region gradually. But if by mistake, if anybody applies high potential to the droplet region, then the physical property of the droplet will be degenerated, and we may lose about the results and outcome of the experiments. Now, when we talk about this microarray of electrodes in the left diagram, then in this particular diagram, the basic concept is how these different cells will be addressed. Because for these particular cells, all these individual cells, we need to send electrical voltage for operating with the electroweighting of diodes. So, talking about the addressing scheme, here we have two different addressing schemes. One is the direct addressing mode, which is the simple one but complex, because in the direct addressing mode, we need to attach control pin with each of these microarray cells. So that's why think about a chip of size, say maybe 24 cross 24 means 24 cells in every row and 24 cells in every column, 36 cross 36. Sometimes it goes to 48 cross 48. So a huge amount of pin is required for this direct addressing mode. So though it's an easy one, but considering the size of the chip, it is quite difficult to fabricate that many amount of pin over there. So later on, it come up with the next concept that is called cross-referencing. This cross-referencing, I guess all of you are familiar about the array addressing scheme, two-dimensional array addressing scheme, which is like the row column addressing. So if you want to address the second row, second column cell, then the addressing is 2, 2. So that will be the second cell of the second row in the two-dimensional matrix. In the similar manner, in cross-referencing mode, we can address in a row column style for each of these cells. And in the direct addressing mode, then think about the pin requirement for biochip, which has m rows and n columns. In direct addressing modes, we require m plus n number of pins. So it is almost nearby order of n square. Whereas, coming to the cross-referencing mode, we require m plus n number of spin only the pins at the column heads and only the pins at the row heads. But still, there are certain actuation issues which need to be taken care about, and if you go through the relevant references, you will come to know that how these different actuation issues can be handled. Now, we were talking about the droplet size in DMMP, digital microfluidic biochip. So these droplet size are specifically nano or picolator of samples. And whenever a bioassay protocol is entered within these particular operations, so each of the droplet movement, they are represented like in the form of either 2-pin nets or 3-pin nets. In the 2-pin nets, which is the left one, you can see one particular location is marked with the S and another particular location is marked with the T. So T is meant by for the target location and S is the source location. So it means a droplet movement over the board, it will begin from the source location and it will go up to the target location following some movement pattern. Whereas for the 3-pin net, we have or we can have two different sources like A and B. They are representing two different source locations. So droplets from two different source locations, they will come to M, which is the mixing region. Over there, they will be mixed and finally they will be moved towards the targeted location. And all these details will fed into the bioassay protocols. Now coming about this droplet movement, as we are now introduced about different nets, and if we look into the droplet movements patterns. Now coming about these droplet movements, there are certain restrictions in this droplet movement. If you look into the left diagram, in this left diagram you can see the droplet, it can move either vertically or horizontally. So there is no diagonal movement. So whenever you want to move droplet from one location to another location, you need to take either the vertical adjacent cell or the horizontal adjacent cell. You cannot go diagonally, because always we know that the diagonal path is this path, which we can traverse from one location to another location in this rectilinear rectangular structure, but we cannot take diagonal path due to certain restriction and its features. So only we need to take either horizontal path or vertical path. In the last slide, it is showing about the electroweighting of dielectric operation. So when you want to move a droplet from one particular location to another location, only your destination location need to be charged with the high voltage or need to be applied with the high potential. But rest of the locations, they should be kept in a ground voltage level. So it is quite challenging in the cross-referencing addressing mode, because you think about one particular row pin or column pin is addressing or it's actuating multiple rows or multiple columns at a time. So there remains some extraneous charge at adjacent cells when we try to activate one particular cell in two-dimensional in row column addressing mode. But if it is a direct addressing mode, then it becomes quite easy, because each of the cells, they are connected with one particular pin, so the direct pin just supply with the high voltage, and for rest of the pin you can keep it into the ground voltage level and it will remain same. Now, whenever a droplet resides into a particular location, this particular thing is known as a critical region. The bar, see the droplet S1, it is residing, and all adjacent cells. You cannot allow any other droplet to enter into this particular cell, because as per the properties of the electrowetting of dielectrics, if any other droplet enters into these adjacent areas, then that will be unknowingly that will be mixed with that particular droplet. So that unintentional mixing of two different droplets might result in some erroneous result. Not mine, it will obviously result in some erroneous result. So always a bounded region, a critical region we need to maintain around the droplet, so that no other droplet by any means enter over there. So whenever a droplet will move from one location to another location, accordingly it's that critical boundary will be shifted from one location to another location. And according to that, in this droplet routing tendency, we formulate the fluidic and dynamic constraints of droplet routing. So whenever we formulate an algorithm for droplet routing, always we need to maintain these fluidic and dynamic constraints. So whatever I have talked about the critical region, the same style, it is the mathematical formulation shown in the right hand side. It tells the static constraints tells that at any particular time instance, no two droplet will reside in an adjacent location. So at any particular time instance, there should be a gap of at least one cell. That is the static. And in the dynamic constraint, it tells that whenever a droplet is residing in any particular location, whether it moves or not, any other droplet should not be allowed at the next time cycle to enter into its critical region. So that is about the dynamic constraints. So now if we look into the basic operation that we need to deal with the digital microfluidic biochip, this basic operation, it deals with dispensing, transportation, mixing, detection, and in addition to this, also we need to take care in the advanced level about the droplet splitting, washing, garbage collection. These all are the part of geometry related operations. And significant researches, if you look into over there, most of the researches, they are targeted or managed with these droplet routing tendencies and droplet routing plans on these digital microfluidic biochip. Apart from that, also we need to take care about the optimal use of control pin or addressing pins. So even if in a row column addressing mode, the number of pins we associate over there, that also need to be optimized. Depending upon different types of bioassay, we need to restrict the use of pins. So the research is going on also in that direction to limit the pins used for routing different bioassay protocols. Apart from that, there is a cross-contamination minimization and residue removal issues related to this. So what is a cross-contamination minimization? Look into this diagram. As we have told that in the digital microfluidic biochip, we can perform parallel operations over there. Parallel operation means parallel droplets can move over the chip. So a cross-contamination can occur in these type of situations where you can see the blue droplets, S and D, by now you know, as you have seen in the two pin nets, that from source to destination it will be moving over there. And for the red droplets, it will move from source to destination. Now, in the shortest path, if we follow over there, In the rectangular marked region, you can see there can be a cell which will be used by both the blue droplets and the red droplets. So irrespective of time, either the red droplet or the blue droplet, any one of them will pass by that location first. So once a droplet passes by any particular location, it's left out some residues over there. So that residue, whenever the second droplet will come, so that residue will be mixed with the second one, and that can result in the erroneous result of or the ultimate outcome of the experiment. So we need to take care of that, and one of the research scope contains this, that how to eliminate these residues, how to read out the plan, and how to, there is a concept of washing operation where we can just circulate the water droplets to move out the regions which has been used by one particular droplet, so that whenever the next droplet will come into that region, it will not be mixed with any leftover residues. So one way of is without washing is the last option which we can follow for this particular situation. In other way, if these cross-contamination issues come, one way is you can manage directly by detour and stalling. In the detour and stalling operation, what we can say, mostly whenever there is a chances of such mixing, which we can see in the first diagram, then what we will do, we will stop the second droplet by moving into the direction where it can mix with another one, and we can detour it. We can find out the next alternative path by which we can reroute it, and by that way we can eliminate the option of mixing. In other way, one is flipping the operation. So flipping the operation means the flipping the path from one particular direction to another direction. In the left diagram, you can see there is a chance of mixing the red droplet movement with the green movement in that particular gray shaded region. So we can eliminate this just by flipping the path of the red droplet movement by the other side of the rectilinear region, so that it can reach to the target by using same amount of electrodes but avoiding the mixing chances. Now we were talking about the pin assignment issues. If you look into this particular diagram, this pin assignment in the cross-referencing mode, the pin assignment is done in this way as we have seen in the left side diagram. So at least to assign 3 consecutive locations in the EMAP movement, we require 3 different pins to address for A, B and C. If we say these are 3 different regions, so at least we require 3 for these 3 adjacent cells, at least we require 3 different pins, P1, P2 and P3. Why this is required? As because if you use A with P1, B with P2, again C with P1, then what happens? We have seen in EWOD mechanism, if your droplet is residing on P2 and you want to move it to the P3 region, say the region C or cell C, then you need to apply a high voltage to the region C. For that reason, instead of using 3 pins, if you use 2 different pins, P1 and P2, then say A and C is marked with P1, then you have to apply high voltage to pin 1. So what that will make, along with making the C region in the charge with the high voltage, it will also pass on a high voltage to the region A. So residing at B, both sides of B, A and C will be charged with the high voltage, and then the droplet will be split from B. It will be split, some portion will be split towards A and some portion will be split towards C, and that will make a trouble. So that's why at least we require 3 different pins to maintain the cross-referencing mode addressing with consecutive addressing. Now if we look into the problem formulation, this is the mathematical formulation. So basic objective of this droplet routing involves at least 3 optimization. The first one is we need to minimize the number of electrodes that is to be used over the biochip for performing one particular operation. And that electrodes minimization, the total electrode minimization is one of the objectives of this droplet routing in digital microfluidic biochip. Next is we need to minimize the overall turn droplet routing. Overall turnaround time means if there are n number of nets, so this overall turnaround says the latest arrival, maximum of the latest arrival should be minimized. Latest arrival means for each of these nets the time requirement. So the net which will take the maximum time among all nets, that should be minimized. So that is the overall completion time that we can minimize over there. And finally, already we have talked about this, we need to minimize the overall income required to emphasize that bioasset protocol. And this is probably the output of this computer optimization technique, that whenever a biochip will be given with certain bioasset protocol, finally we write this kind of a routing map where the black shaded regions are the block cells, they may be faulty or there may be some on-chip preserver placed over there. So from the rest of the available areas, we need to route different droplets together. And all this routing has to maintain those critical regions and other constraints of droplet routing. So in the design automation, still you can explore like the application of swarm-based method for efficient droplet routing, because mostly the techniques which has been used for this droplet routing mechanism, they are based on graph theoretical methods, integer linear programming, incremental analysis and the whole. So specifically the application of intelligent mechanism and the swarm intelligence methods are not in that scale, maybe due to the complexity of the overall nature of the problem. Next is contamination minimization, synthesis of interlinked bioasset protocols, that is also another scope of research area. Wind constant droplet routing is another scope, and wind co-optimization. Also the module synthesis, module placement during execution of bioasset protocols can be considered as prospective areas for research and design automation of this. Now coming to the last section of this lecture, that is the fault management. So if you look into the reason for the fault occurring in the biochip, they can be classified as into these 5 different categories. The first one is the dielectric factor, which occurs for the excessive actuation voltage applied to any particular electrode. This excessive actuation voltage applied to any particular electrode, it can result for 2 different reasons. One reason is you are utilizing one particular electrode or a couple of electrodes together repeatedly for different bioasset protocols. So repeated use of certain number of electrodes for the bioasset protocols in compared to others will automatically degrade the quality of those electrodes which is repeatedly being used in compared to others. Otherwise, there may be some erroneously you can pass on some high voltage to a particular electrode due to the failure of your electron voltage control mechanism, and that will also degrade the quality of electrode. Next reason is irreversible charge concentration, that is that any electrode you kept it charged for a prolonged period of time. Misalignments of parallel plates, as you can see there is a very small gap between 2 different cells, like in the region of 0.4 micrometer to 0.2 micrometer. So certain misalignment, it will be 2 different adjacent cells will be overlapped with one upon each another, and that will cause an erroneous result. Next one is the non-uniform dielectric layer. So overall depositions of dielectrics, as it is based on any miniaturized level you can perform in this. So uneven distribution of dielectric components over there will cause results, and sometimes the grounding failures also will cause results. So fault management procedure means whenever your biochip has encountered any fault, and it is not like the whole chip is under fault region, that occurs, seldom it occurs. Quite often what occurs is that some of the microelectrode arrays has been affected due to this fault reasons. And if some of your electrode arrays are affected, then to handle those fault scenarios, there are 2 different ways. The first is, you can see the fault location are already identified. You know that what are the electrodes which are faulty. So you can design a routing map just by bypassing those electrodes. You just can avoid those electrodes. So the active electrodes you can use, and easily you can derive a proper routing situation. But sometimes there might occur a situation like the number of active electrodes available is not sufficient enough to route all of your bioassay protocols. So in that case, you need to take a biochip board. And another research, which is a complex one, that encompasses both fault identification. In the first case, fault locations were known to us. In the second case, you need to find out what are the microcells as met with the faults. And then, along considering that fault, you need to perform a fault-aware routing. What is the fault-aware routing? You can see in this diagram that suddenly you came to know the red region, it has encountered with the fault. So whenever you route your droplet, you need to bypass it, if possible. And this is the fault identification mechanism used. Commonly we use like by parallel scan or distributed scan mechanisms are there, where we need to send the test droplet first. That's because all the reagent and the samples are costly. So first we need to pass on the test droplet, which is mostly the water droplet with the potassium chloride solution, KCl solution. So you need to pass on that, and that droplet passing, you first need to circulate either column-wise or row-wise. Over here it is shown like the row-wise circulation. So row-wise, whenever you pass on the test droplet, still you maintain the critical region for the test droplet also. So you can see first the alternative rows are being used for scanning and circulation of droplets. Then in the next term, you can see the rows which were initially used, then the next adjacent rows are being used for scanning. So this is first the row-wise scanning, and then you can do the parallel scanning, that is the vertical scanning. And now by taking the cross of that row-wise scanning and the vertical scanning, you can identify the actual regions of the cells which has met with the errors. But again, this fault identification, this is a very crude method. You need to optimize the time, because in between two different bioassay protocols, you cannot spend too much time identifying these faults. With that, you need to optimize the time, and you need to find out a technique by which you will be able to detect the fault location with minimum possible time. So the accuracy in the results of DMFT is important. So mostly different researchers of DMFT are mostly focusing on design synthesis and fault-aware operation. Many open areas are there, open problems like defect-aware pin construct routing, defect-aware cross-contamination detection, residue removal. You can address and you can explore your research concept over these. Different reasons for faults are known to us, but we need to focus on certain mechanisms by which we can minimize this reason to occur for a particular digital microfluidic biochip board, so that the frequency of fault operations can be minimized. So with these certain bibliographies that we have referred for preparing this lecture, and mostly you can see in the Duke University there is Professor Krishnendu Chakravarti. You can refer to the publications of Professor Krishnendu Chakravarti from Electrical Engineering Department of Duke University. You will come to know about many details of regarding this DMFT and its recent publications and work trends, and you can go through these research papers which are mostly available on different IEEE export and different IEEE proceedings. And with this, we end with this session of keynote address on digital microfluidic. Thank you. Question?
Next, Dr. Shubhra Sankar to kindly present a memento to Dr. Indrajit as a token of appreciation. It's time for tea break. I would request